Okay, we're going to go ahead and get started. So the first thing I want to mention is hopefully I'll have recordings of our sessions up this weekend. I've been trying to start a Facebook page, and for some reason I've been kicked off Facebook. I can't figure it out because I've really never made any posts. I've told a few friends happy birthday and things like that, but and YouTube is harassing me because apparently our videos are too long. So, but I should have either a YouTube or a Facebook spot where we can go to if you want to view the videos again by the end of the weekend. I've got somebody from the IT department at school helping me out. I think we'll be able to figure it out. We're going to start out by looking at the problems from yesterday that I asked you to look at home. So I'm going to go to screen sharing and we'll look at those. And we're going to start out looking at our population of deer. So keep in mind what I said yesterday, which is, in these cases, we're actually having to count the numbers to show what the frequency of the dark phenotype and the light phenotype is. But a lot of times those will be given to us in the problem. And we won't have to count, we'll just get a frequency, or we might get a number, 200 people out of 280 people, something like that. So what's the frequency of the little d allele? So when I looked at the population, I counted 8 light-colored deer out of 50 total. So if we take 8 over 50 and I reduce that fraction, so that's 4 over 25. And notice that's the frequency of little d squared, because that's deer that show the light phenotype. So to have that phenotype, you have to have 2 recessive alleles. So your little d, little d. So remember what I said the other day, this is why it's crucial to think in terms of associating big D, little d to different combinations with genotypes and phenotypes. So if little d squared is 4/25, we can take the square root of that, and little d will be equal to 2/5, or if you're doing decimals, 0.4. And again, notice only 8 out of 50 deer show the recessive characteristic, but the frequency of the recessive allele is 0.4. So it's almost half of the alleles in the population. And again, this goes back to what we said. Why don't you see more light-colored deer? Because most of those recessive alleles are in heterozygotes, and the dominant allele is what's going to show up in a heterozygote. And remember, that's why we have to start with the light-colored deer, because we know their genotype. So what about big D? Big D is going to be 1 minus little d, or 1 minus 0.4, so 0.6. Or if you were using fractions, 1 minus 2/5, 3/5. So whichever you'd prefer is fine with me. If this population is in Hardy-Weinberg equilibrium, what'll be the frequency of the little allele next generation? Well, remember, in Hardy-Weinberg equilibrium, the allele frequencies stay the same from one generation to the next. So it's going to be 0.4 again. What's the frequency of the big D allele? 0.6, because again, if the population is in Hardy-Weinberg equilibrium, things stay the same from one generation to the next. If the population is in equilibrium, what's the frequency of little d genotype? So that's 0.4 times 0.4, that's 0.16. What's the frequency of big D, little d genotype? Or notice another way this question could be worded, what's the frequency of heterozygotes in the next generation? Well, even though it says what's the frequency of big D, little d, remember our Hardy-Weinberg equation, you've got to have that 2 out in front of big D, little d. So it's 2 times 0.6 times 0.4. And if you multiply that out, that's 0.48. Again, if our population is in equilibrium, what's the frequency of big D, big D? 0.36, 0.6 times 0.6. Now notice, if you think about question 5 and question 6 and question 7, these are the only 3 genotypes that are possible in this population. So one way we can double check our work at this point is if we add all these up, they should add up to 1. Or alternatively, for this last one, oops, we could have said that big D is going to be equal to 1 minus little d, and also minus 2 big D, little d. Oops. So in other words, we could have gotten the answer to 7 by subtracting 1 minus 5, and then whatever our answer is there, subtract our answer for 6. And that would be 1 minus 0.16 minus 0.48. And that does give us 0.36 again. So a lot of times there's more than one answer mathematically to come up with, more than one method, I should say, or way to come up with the same answer mathematically. I don't really care which method you use. I do like you to kind of show your formula at the beginning so I can see if you're on the right track. And when we have some questions about this, keep in mind that these questions get graded kind of like in a math class. So you do get partial credit if you have the right formula and then you substitute in the wrong numbers or you multiply wrong or do something like that. But if you don't have the right formula, you lose some credit. So usually when these questions are on the question sets that I grade, you know, a question like number 6 might be worth 3 points. So you'll get 1 point on this question for having the right generic formula. You'll get 1 point for showing me the numbers that you substituted in. And then you get 1 point for getting the correct answer for actually doing the math correctly. If the population is in Hardy-Weinberg equilibrium, what will be the frequency of the dark phenotype in the next generation? Well, we can find the dark phenotype just by counting. And when I counted, I got 42 dark colored deer out of 50. So if we convert that to a decimal, that's 0.84. What will be the frequency of the light colored population? And again, you could count. If you count, you get 8 over 50. But the other way you could do this is you could say, you know, the light color is equal to 1 minus the dark color. So the dark color is what we just figured out in number 8. So we can take 1 minus 0.84. Whoops. And I subtracted the wrong number, which is why it's a good idea to write this out and then double check your work, as I didn't do in that case. Are there any questions about how we got these answers? Okay, let's move away from this screen and let's look at our natural selection problem. So I'm going to go to screen sharing again, but I'm going to go back to our diagram we had with our peppered moss. Let's try again since it doesn't seem to want to do that. Okay. Oh, that's why. Skip down. Okay, I think you can see problem A, the problem that we were working on yesterday in class. If that's not what you're seeing, let me know and I'll double check and make sure I'm sharing the right screen. So what I did in class yesterday, we worked a few of these and we went through and we counted up the totals for each of the rows. So what I did in each case was I subtracted, or I divided, I should say, the number in a cell by the total for all the cells in that row. And I carried my answer out to 3 decimals. When we work these problems, we'll usually be carrying our answer out and rounding to the 3rd decimal, maybe the 4th, depending on the problem. And then I did the same thing after I did the totals before selection. I did the same thing to the totals after selection and the totals selected. And then what I did with each one was I compared the frequencies selected and the frequencies after selection with the frequency before. So if we look at number 1, the frequency before selection was 0.38. So I compared that to the frequency selected, which was 0.36, and this number is lower than the frequency before. I compared the frequency after selection, which was 0.039 to 0.038, and this was higher than the frequency before. So I went through and I did this for all 9 color categories. And notice, and it makes sense if you think about it, if the frequency selected is lower, then the frequency after selection is higher. If the frequency selected is higher, then the frequency after selection is lower. Because basically what this means, remember we said the frequency selected is the individuals that are being killed by natural selection. In this case, they're being eaten by birds. If more of them get eaten by birds than you would expect, then that means the frequency is going to decline after selection. Now, keep in mind, whenever we say, what do you expect to happen? What we really expect to happen is the exact same thing as these frequencies before selection. So if this is our population before selection, if 0.038, if that's the frequency of the number 1 color before selection, we expect that to be the same frequency that's selected. And we expect that to be the same frequency after selection. And we expect for the number 2 color, since it's 0.086 before selection, that should be the frequency that is selected. And that should be the frequency after selection. Now, why do I say that we expect them all to be the same? Think of reaching into a grab bags. If you have half red marbles and half green marbles and they're in a bag, your chances of pulling out a red marble or a green marble are a half for each color. So here, 38 out of every 1,000 moths are number 1 color. So if we reach into a big grab bag of moths, about 38 times out of 1,000, we ought to pull out a number 1 color. Now notice, if our frequencies would match up perfectly, if the frequency selected and the frequency after selection are exactly the same as the frequency before, what does that mean? That means there's no natural selection at that spot. You know, we could see a case where there's no natural selection, where it doesn't matter what color an animal is in terms of whether it gets eaten by a predator or not. It could be that there's something else in play. So if this number is lower, this number is higher. Now, let's look at the 2 questions that go along with each chart. So were moths of all colors picked in equal numbers? Well, notice when it says picked, that's the selected. So that's our column that says, or our row that says total selected. So it says, were they picked in equal numbers? So we're not asking about frequencies there. So we're going to say all colors were not picked in equal numbers. Were any moths picked more frequently than would be expected? Okay, so more frequently than would be expected. What that is asking is, are any of the moths picked at a higher frequency than would be expected? Well, yes, 4, 5, 6, 7, and 9 are all picked at a higher frequency. Now, if we go back, we're going to pick less frequently than selected. These would be the ones where the frequency selected is lower. So if we go across and we look, 1 is lower, 2 is lower, 3 is lower, and 8 is lower. Okay. So now the next question is, does this show directional selection, disruptive selection, or stabilizing selection? Well, remember when we looked at the 3 type, in directional selection, moths at one end have a higher chance to survive. Moths towards the other end have a lower chance to survive. If it's disruptive selection, moths in the middle have the lowest chance to survive, and moths at both ends have a higher chance to survive. And if it's stabilizing selection, moths at both end, the high end and the low end, have a lower chance to survive, and moths in the middle have the highest chance to survive. Well, if we look, notice 1, 2, and 3 all have a higher chance to survive. After we get past 3, almost everybody has a lower chance to survive. Number 8 is a little bit of a blip. It goes up slightly. But because 1, 2, and 3 all have a higher chance to survive, and then the other end, 5 out of 6 of them have a lower chance to survive, we would say that this is directional selection. Why? Because moths at one end have a higher chance to survive. Oops. Moths towards the other end. And notice we're repeating our numbers here again. Now notice, you might think, okay, well, number 8 completely tears down the idea of directional selection. But keep in mind, when people design ways to classify natural selection like this, just like when people do anything where we divide nature into categories, nature doesn't always cooperate perfectly. So this is definitely predominantly directional selection. Now, number 8 is a blip. Maybe number 8, remember we said these moths hide on tree trunks. Maybe number 8 actually hides on a flower and happens to blend in with that flower or something like that. Maybe there's another explanation for why that's kind of an exception to the rule. But we would definitely say that this is directional selection. Now notice, if you were to make a prediction about this population for the future, you would predict that in the future, on average, moths in this population are probably going to be a little bit lighter in color. Why? Because the lighter colored moths are having a higher chance to survive. The darker colored moths, again, with that one exception of number 8, have a lower chance to survive. So if this happens, if this pattern continues for many generations, then probably the lighter colored moths are going to become more common. The darker colored moths are going to become rarer. And this is what happened when pollution controls were put into effect in the 60s. Now, if we were looking at some numbers from the time when the Industrial Revolution began and pollution started making tree trunks darker, we would still see directional selection, but then it would be the darker colors that would be more likely to survive, and the lighter colors would be less likely to survive. Now, if it had been moths towards the middle, if it was 3, 4, 5, 6 that had a lower chance to survive, that would be disruptive selection. If the two ends are more likely to survive and the middle is less likely. If it was stabilizing, moths at both ends would be lower in terms of their survival possibilities. So notice there's 2 basic ways to make your comparison. You can compare before selection with after selection, and you have to compare frequencies. You can't just compare raw numbers. That won't give you the correct answer necessarily. But if you compare the frequency selected with the frequency before, if this number is lower, that means the moths in that group have a higher chance to survive. Why? Because think about what this number represents. This is the moths who got eaten by birds. So if this is lower, these moths were better camouflaged. If this number is higher, that means more of these moths got eaten than we might expect. So they're less likely to survive. Notice that the higher and lower is reversed when we get down here, because here we're looking at who got eaten when we look at frequency selected. When we look at after selection, we're looking at who didn't get eaten. So if one of these is higher, the other is going to be lower. Are there any questions about how we would work a problem like this? Oh, I guess I forgot another pattern of, well, it's not really a pattern of natural selection, but we talked for a moment about disruptive selection or stabilizing selection. And notice there is another pattern you could see here. The other pattern you could see is what I talked about briefly before, no natural selection. What if when we looked across the frequency selected, that this was 0.38 in the first frequency, and it was 0.38 in the frequency selected and 0.38 below. And, for moth category number 4, the frequency before selection is 0.163. If the frequency selected was 0.163 and the frequency after selection was 0.163, then if the frequencies don't change, if they're the same in all 3 categories, then there's no selection, because that means everything gets picked exactly as often as it's represented in the population. If a third of the moths are number 1, a third of the moths eaten are number 1. And when you get done, a third of the moths left will still be number 1. So we want to keep that in mind. So we could see one of 4 patterns here. We could see directional selection, like this example. We could see disruptive selection, which is sometimes also called diversifying selection. We could see stabilizing selection, or we could see no natural selection. Now in nature, no natural selection is very rare, but we do see it on a few occasions. But there's almost always something. But the reason we typically might see a chart that shows no natural selection, suppose we're looking at moths where colors have no bearing on whether they get eaten by birds or not. Maybe there's another factor that affects whether they get eaten by birds. Maybe it's their wingspan. basically how big they are. Bigger moths are sometimes easier for birds to see than smaller moths. So in that case, we might look at a chart of the colors and we might see no natural selection because, you know, color is not what affects their odds of survival. So are there any questions about this sort of problem? Okay. I'm going to stop sharing this screen then, and we're going to go back to some of our notes that we looked at the other day. And we're going to continue with those. So remember when we left off in our notes, we were talking about factors that could affect Hardy-Weinberg equilibrium. And basically when these factors are operating, Hardy-Weinberg equilibrium doesn't occur. And we said that in real populations in nature, most of the time, Hardy-Weinberg equilibrium is not in effect. So the last topic we were discussing was different aspects of genetic drift, which is sometimes called random genetic drift or sometimes called genetic sampling error. And basically genetic drift is something that's important in small populations. It's not crucial in large populations. So when we think about genetic drift, what we want to think about is in nature, what conditions create small populations. So we mentioned one the other day, the founder effect, where one or a few individuals get separated from the main population and start a new population. And when that happens, then this new population, its gene pool, the allele frequencies, that's going to depend on the individuals that were in that founding population. So here's an example showing one individual getting separated. Now you might ask how one individual could start a new population, but there have actually been some documented cases where one individual got separated from a larger population, and it was a female who was carrying eggs. Or some species have both male and female reproductive organs, so they can reproduce on their own. So there are a couple of ways that even a single individual might start a new population. The other common situation that we see in nature that can produce a very small population is something called a population bottleneck. So think of representing our population by putting them into a bottle. The bottleneck is the top part of the bottle, the part that's narrower. So in the bottleneck effect, what happens is we have a large population represented by all these little red and yellow circles. And those are kind of our two phenotypes in the population. And something drastic happens and the size of the population is reduced dramatically. So we go from having a population represented by all these little circles in the bottle to just a few circles over here. Well, it's very similar to the founder effect. So which individuals survived this drastic event? Those individuals that survived this event are the individuals that form the basis for the new population. Why? Because everybody else is wiped out. Remember when we talked about Darwin's ideas, we talked about one of the stops that the Beagle made on its round the world voyage was in the Galapagos Islands off the coast of Ecuador. And since Darwin's visit in about the last 100 years, maybe not for about the first 40 years after his visit, but In the last 100 years, a lot of biologists have studied the populations on these islands. And a good example of the bottleneck effect is the one type of bird on the island. There's a large population of birds. But on this island, there was an extreme drought for 2 years. And in the second year of the drought, the bird population there typically lays 2,000 or 3,000 eggs every year. The second year of this drought, only 2 eggs hatched. All the other eggs died. So think about this population in the future. This population in the future is going to be derived mostly from these 2 individuals. Now, birds live more than 1 year, so the number of eggs that hatched the year before and the year after also had an impact. But that's a good example of a population bottleneck, where instead of hundreds or thousands of eggs hatching each year, 2 eggs hatched. So in both cases, we have a new population that's formed from just a few individuals, whether we're talking about the founder effect, where the new individuals move to a different area away from the main population, or the bottleneck effect, where most of the main population doesn't survive some genetic event. Now, another characteristic that can change allele frequencies is something that we usually just generically refer to as genetic recombination. And recombination means new combinations of alleles. Now, it's not exactly like mutation, because a mutation can create a completely new allele. But what genetic recombination does is it creates new combination of alleles. And there are a couple of ways that this can happen. The most common way, and a way that we talked about back in Biology 105, was a process called crossing over. So here we have a representation of 2 homologous chromosomes at interphase 1, at the end of interphase 1, after the DNA has been replicated, or prophase 1 during meiosis. So on one of the homologous chromosomes, we have 3 alleles, big A, big B, and big C. On the other homologous chromosome, we have 3 alleles, little a, little b, and little c. Now remember, without crossing over, this organism would pass down either the sister chromatid that has the big A, the big B, and the big C, or the sister chromatid that has the little a, the little b, and the little c. But we can have crossing over. And remember, in crossing over, when the chromosomes get tangled up, they get reattached, but they get reattached to the wrong sister chromatid. So after crossing over, now we have big A, big B, and little c on the same chromosome. So those 3 alleles are going to be inherited together. We have little a, little b, and big C. So those 3 alleles are going to be inherited together. And you might think, why does this matter if the alleles haven't changed? Well, as we've learned more about genetics, one of the things that we've discovered is, of course, it's always the case in biology, things are not as simple as they might seem at first. We used to think that this A gene, whether you get the big A allele or the little a allele, that protein does its thing and it doesn't matter what else is going on in the cell. And if we look at the B gene, you get either the big B or you inherit the little b allele. And again, that B protein works the way it works with no outside interference. But what we know now from studying metabolism and biochemistry is that one protein can affect the way another protein works. So if you get little c being made at the same time as big A and big B, that may give you a completely different result than when little c was on the same chromosome with little b and little a. So even though the genes haven't changed, the phenotype that you get from the genes might be different. So that's not necessarily the same. And that's why genetic recombination is important, because when we get these new combinations of alleles, sometimes we get a new phenotype. proteins in your cells are not independent entities. They're affected by other proteins in their environment. And another reason is, remember, we also talked about in Biology 105, some of the areas on the chromosomes are actually on-off switches that tell the cell to make a protein or not to make a protein. So you could potentially have something moved from an area where it was under the influence of an on switch in the DNA to an area where it's controlled by an off switch. So the cell isn't going to make that protein anymore. So as we're learning more about genetics, as I say, we're discovering that it's not as simple as we would like it to be. And finally, the last process that we're going to talk about, keep in mind there are one or two other processes that can occur, but they're very rare and seem to be minor, is something called sexual selection, or it's also called non-random mating. And sexual selection produces what is commonly called differential reproductive success, meaning individuals produce a different number of offspring. Now, notice natural selection is often referred to as differential survival. And the reason that sexual selection is often talked about differently is because being able to reproduce successfully is not necessarily tied to being able to survive. In Darwin's original book that he wrote in 1859, he kind of ignored reproduction. He basically focused on survival. And later, he realized that he had ignored reproduction and it was important. And he wrote another book about the same size on sexual selection. And again, like natural selection, it turns out that this is a hypothesis that can be tested experimentally. And sexual selection has been studied in a lot of animals, a lot of plants, and fungi. And you might think, you know, sexual selection couldn't work except in animals, but it does. And the basic idea of sexual selection, we'll look at it in a little more detail, but the basic idea, notice that this is also called non-random mating. So the basic idea is this is different than random mating. You know, if mating was random in something like people, for instance, then males and females would get together with no apparent pattern. Yet, you know, when we study this in sociology, we know that people tend to have children with people who are similar to themselves from the same ethnic, racial, social, economic, religious, educational background perspective. Now, it doesn't always work that way, of course, but when we talk about non-random mating, we're talking about individuals mate with other individuals, and it's not just anybody. They, in some sense, choose who they mate with. And sexual selection really occurs because of one main factor. In almost all organisms, females invest a lot more resources in reproduction than males do. And when I say resources, think about an egg cell and a sperm cell. Egg cells are much, much bigger than sperm cells in almost all living organisms. In people, an egg cell is about 2 million times larger than a sperm cell. And in fact, really all the sperm cell contributes to the egg is the 23 chromosomes from the father. All the cytoplasm, all the organelles in the cytoplasm come from the mother in the egg cell. There are some exceptions to this. There's one type of cricket where male crickets make sperm cells that are about 1/4 of their body weight. Now, here's what we want to keep in mind. Think about this like an economic proposition. So I just said that, you know, in humans, an egg cell is about 2 million times as expensive, if you want to think of it that way, as a sperm cell. So suppose we were looking at a business partnership, and so I'm going to proposition one of the young ladies in the class, and suppose I said, okay, I want to start a business with you, and here's the deal. I'm going to invest $1 in the business and you invest $2 million. And if the business is successful, I'm willing to split the profits with you 50-50. Doesn't sound fair, right? But think about how reproduction works. Females invest more resources than males, but if reproduction is successful, they both pass down half their DNA to the offspring. So they both get half the profits in a sense. So if you think about sexual selection as kind of a business strategy, well, since females invest many more resources, females want to make sure they choose a good business partner. Whereas males, they just want to start a business. So let's look at the cost of reproduction and why it's higher for females. In a lot of species, males produce sperm continuously, and females are much more limited. Not just in people, but actually in most animals that we study, females spend the most time on parental care. So after offspring are born, the male might go visit another female, but the female is kind of stuck at home guarding the nest or the den. So because females invest more, the ultimate result is females are picky. They're gonna do this. So we basically see two types of factors that play a role in sexual selection. One is intra-sexual selection. Now, intra means within. So this is competition within one sex, and it's almost always males. And this says to meet, but really probably it would be better if this says to mate with members of the other sex. So intrasexual selection typically involves a couple of things. It involves threat displays, trying to scare other males away. If you would look at two male deer, when they're fighting over a female, they kind of pace back and forth. They're parallel to each other. And then before they would actually fight, they actually turn and face each other. And when they turn and face each other, the two deer can see the spread of the antlers. And if your antlers are bigger, okay, that's probably a bigger deer, not always, but so the idea is to scare the other male into backing down. Why? Because if threat displays don't work, in a lot of species, the next thing that happens is combat. The males fight. And you know, the reason a threat display is better than combat, if you have to fight, you could get hurt. So the idea of combat is, and you've probably seen on some nature special, the two deer kind of jousting with their antlers. Or you may have seen sea lions where they have an area on a beach, and when they fight another male sea lion, they kind of try to push each other around like sumo wrestlers. Now, another way that males try to control access to females is through a process called territoriality. A male stakes out an area and then defends this area. And females that come into this area, that's his harem, that's his group of females. And if another male tries to sneak in, the male will fight that male. There's also something called sperm competition. When we think of sperm, we think of cells that are designed to fertilize eggs. But remember, I told you that I studied reproduction in insects. And when we look at insects, they have some incredibly unique ways of reproducing. Some insects make two types of sperm. So when a male insect ejaculates into the female, some of his sperm are sperm that fertilize eggs, but some of his sperm are cells that are similar to cells in our immune system. Well, cells in our immune system attack foreign cells. So some of this male sperm will attack foreign sperm. So what's the idea? If the female's mated with somebody else, kill off those sperm before there's a chance for those sperm to fertilize the eggs. Some male insects have a penis that has two parts to it. One part delivers the sperm, but before they deliver the sperm, they put in the other part of the penis, and the other part of the penis works like a scoop. And they scoop out any sperm that's already in her reproductive tract. Again, eliminate the competition. Some males, when they finish reproducing with the female, the sperm hardens in her vagina and forms what's called a vaginal plug. So she can't mate with anybody else. Except in a lot of these species, the penis has a little attachment that's a corkscrew that they can remove plugs with. And finally, even after a female mates with another male, in a lot of species, the males practice infanticide. They kill the babies. You know, probably a lot of you saw that Disney movie, the Lion King. And the Lion King was a very sanitized version of this. If you would look at a pride of lions in Africa, a pride of lions is a bunch of females, usually sisters or cousins, and their cubs. Female cubs stay in the lion pride. Male cubs usually get kicked out by about the time they're 2 years old. Now, besides all the females in the pride, there's one male. Every once in a while, two males who are brothers, but almost always just one. And when the cubs get kicked out of a pride, they wander around on their own. And eventually, if they don't die, they get bigger, they get stronger, and they'll challenge another male for control of a pride. And if they can defeat this other male, and this does involve real combat, if they can defeat this other male, that male's kicked out. And the new male that comes in, the first thing that he does, he kills all the cubs under 2 years old. Why? Because until the cub is a couple years old, the female spends a lot of time taking care of the cub. She doesn't come into heat again. She's not sexually receptive. So he kills all the cubs so that the females will mate with him. And they have a short memory, they mate with him, but now the cubs are carrying his DNA, not his predecessor's DNA. So the Lion King kind of approached this, but as I say, in a very sanitized version. So here's one example of male competition where we have some things called stag beetles. And stag beetles have a big, almost like an antler on the front of their head. And basically they use this lever kind of like a pry bar. And when males are fighting, they typically fight over tree branches. They try to get their horn underneath a male and they flip them off the tree branch. Now, keep in mind, insects are very light, so they can fall 30 or 40 feet without getting hurt. So the other male isn't killed. But the reason they do this is because females will lay their eggs on an area of a woody tree branch. And the females mate right before they lay the eggs, literally a few minutes before they lay the eggs. So if a male can throw an opponent off, now he has this little tree branch that he can patrol and mate with any females who happen to light here, who land. Now, intersexual competition, keep in mind, intra means within. So intrasexual competition is mostly within males. And since males make a lot of sperm and females don't make too many eggs, it's usually males fighting for the right, for the chance to mate with females. Inter means between. So intra is between members of one sex, usually males. Inter is between members of different sexes. So this includes males attempting to attract females and then females choosing which males they'll mate with. So how do you attract females? Well, one way is by holding a branch, holding a territory where a female will lay her eggs. In many species, a female won't mate with a male who doesn't hold the territory. Now, what are females looking for? Well, a couple of things. Can the male provide good resources for the offspring? Notice that on the right side, this is actually looking at some of the attempts to apply sexual selection to people. And there is some indication that there is sexual selection in people, but not quite as simple as in some animals. So what are good resources? Well, there are some insects called Dobson flies. And for a female to mate with a male, the male has to, Dobson flies are predatory, they kill other insects. The male has to kill her a fly and bring her a nice juicy fly to eat. And Dobson flies aren't very romantic, so what happens is, while the female is eating this fly, she'll let the male mount her and reproduce, transfer some sperm. So if you bring the female a nice big juicy fly, it takes her longer to eat it. You get to transfer more sperm, fertilize more eggs. If you bring her kind of a shriveled, dried up small fly, it doesn't take her long to eat it, you don't get to do much mating. So it could be good resources for the female, but it could also be good resources for the eggs. Most female robins will only mate with a male who holds a territory. And that territory is where the robins can collect food to feed their hatchlings. Now, we'll finish up talking about females on Monday, but it's time to go. But before we go, I would like you to work problem B on the natural selection problems. So I'll send out an email about that, but please work problem B. So you're going to work it similar to the way we did. You're given the numbers before selection and the numbers selected. So you're going to have to subtract to find the numbers after selection. And then you're going to calculate the frequencies. So this will be the last practice one, and then we're going to work one or two that you'll have to turn in. So work this, see if you can master it. If you have any questions over the weekend, send me an email. But otherwise, have a good weekend, and I'll see you Monday morning.
See you Monday.
Bye-bye.
Thank you.
Weekend.
Enjoy the weekend. Looks like maybe at least today there will be a little bit of sunshine, but maybe a lot of snow by Monday, I hear.
Yeah, maybe tomorrow it will snow.
Well, hey, it's good to get out and shovel a little bit. Get some exercise instead of just sitting in Zoom sessions all day.
Mr. Bishop, I didn't see the attachment. You didn't get to attach the file. You didn't attach.
Yeah, it said the attachment was too big and it wouldn't let me put it on. So I'm gonna try from a different email address, and if not, I'm gonna see if I can get the website up and running this weekend, and then they'll all be up there.
Thank you.
You're welcome. Have a good weekend.
I was just worried because
No, you weren't going crazy. I meant to send you another email saying it won't let me add the attachment. It keeps saying it's too big, and then I forgot about that.
Yeah, because you know what, classes, once you start having works to do now, going back would be very difficult, you know.
Yeah.
Yeah. So thank you very much.
We'll figure something out. I hope to have everything up before we meet on Monday, and if I do, I'll send you an email and let you know what URL you have to go to or how to access it.
Right. Thank you very much. I really appreciate it.
You're welcome. Have a good weekend.
You too. Bye.
Bye-bye.